हाय गाइस, वेलकम बैक टू माय चैनल। तो आज हम डिस्कस करने वाले हैं क्लास 12 एनसीईआरटी बायोलॉजी चैप्टर 5 मोलिकुलर बेसिस ऑफ इनहेरिटेंस। ये चैप्टर नीट के एग्जाम के लिए बहुत ही वेटेज रखता है। सबसे ज़्यादा क्वेश्चन इस चैप्टर से ही आते हैं नीट के एग्जाम में, और सबसे ज़्यादा यही चैप्टर नीट एस्पायरेंट को टफ लगता है। तो गाइस, यहाँ पे हम सारे के सारे इंपॉर्टेंट पॉइंट, इंपॉर्टेंट टॉपिक कवर करने वाले हैं कम से कम समय में। तो चलो स्टार्ट करेंगे। द डीएनए का लेंथ डिफाइन किया जाता है नंबर ऑफ न्यूक्लियोटाइड से। बैक्टीरियोफेज नॉन एज द 5 × 174 हैज़ 5386 न्यूक्लियोटाइड एंड बैक्टीरियोफेज लैम्ब्डा हैज़ 48502 बेस पेयर। Escherichia coli has the 4.6 × 10⁶ base pair. Haploid content of the human's DNA is 3 × 10⁹ base pair. DNA is an acidic substance present in the nucleus was first identified by Frederick Messer in 1869. He named nucleins. Elucidations of structure DNA remain elusive for very long period times. Only in 1953, James Watson and Frederick Creeks, based on X-ray differentiation data produced by the Messer, Wilking and Rosalind Franklin, proposed very simple famous double helix model for structure of DNA. One hallmark of their proposition was base pairing between the two strands of polynucleotide chains. This proposition was also based on the observation of Ivan Chargaff that for double-stranded DNA. Silent feature of the double helix structure of DNA are follows. First, it is made of two polynucleotide chains where the backbone is constituted by the sugar phosphate and base project inside. Second, the two chains have antiparallel polarity. It is means if one chain has a polarity 5 to 3 directions, the other is 3 to 5 directions. Third, the bases of in two strands are paired throughout the hydrogen bound edge bound forming base pair adenine from two hydrogen bound with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine and 3 H bonds. As result, always purine comes opposite to pyrimidine. This is generated approximately uniform distance between the two strands of helix. Fourth, the two chains are coiled in the right-handed fashion. The pitch of helix is 3.4 nanometers on the billions of the meters, that 10⁹ meters, and there are roughly 10 base pair in each. Francis Crick proposed the central dogma in molecular biology, which states that genetic information flow from DNA, RNA, protein, replication, DNA, transcription, mRNA, translation, protein, central dogma. In some viruses, the flow of the information in reverse direction, that is from RNA to DNA. Packaging of the DNA helix, two consecutive base pair as 0.34 nm, 0.34 × 10⁹ mm. Length of DNA double helix is typical mammalian cell is calculated simply by mapping total number base pair, that 6 × 10⁹ base pair into 0.34 × 10⁹ meters base pair. एंड डीएनए का अप्रोक्सीमेटली लेंथ होता है 2.2 मीटर्स और टाइपिकल न्यूक्लियस अप्रोक्सीमेटली 10⁶ मीटर्स. In prokaryotes such as E. coli, throughout they do not have defined nucleus. The DNA not secretes throughout the cell. DNA begin negatively charged. In eukaryotes, positively charged basic protein called histone. Histone are rich in the basic amino acid lysine and arginine. Histone are the originated from the a unit 8 molecule called histone octamers. The negatively charged DNA is wrapped around the positively charged histone octamer to form structure called nucleosome. Typical nucleosome contain 200 base of DNA helix. Nucleosome constitute repeating unique structure in nucleus called chromatin. Thread-like strand colored body seen in the nucleus. The nucleosome in chromatin are seen as based on string. The based on string structure in chromatin is packed to form chromatin fiber that are further collated and condensed metaphase stage. Non-histone chromosomal protein. A typical regions chromosome are loosely packed and strand light are referred to as euchromatin. The chromatin that more densely packed and strand dark is called heterochromatin. Euchromatin is said to be transcriptive actives and chromatin were heterochromatin inactives. The search for genetic material, discovery of nucleus by the Ncert and proposition for the principle of inheritance by the Mendel were almost the same time, but DNA acts as genetic material discovered and proven by 1926. For genetic genetic inheritance and reach molecular level. Previous discovery by the Gerge Mendel and Walter Sutton and Thomas Hunt Morgan. Transforming principle 1928 Frederick Griffiths. Serious experiment with the Streptococcus pneumonia visit us the microculous transformation in the bacteria. S10 bacteria have the mucous polysaccharide cut. S10 does not. Mice infected S10 virulent died from the pneumonia infection, but mice infected with R10 do not develop pneumonia. S10 injected into mice died हो जाएगा. R10 injected into mice live, and heat killed S10 bacteria injected into mice did not kill them. S10 heat killed injected to mice lives, and S10 killed plus R10 live injected into mice died. Injected a mixture of the heat killed S and R bacteria, the mice died. Biochemical characterization of transforming principle. Oswald Avery colonized MSCLDO and Malsin Messi-Scrati 1933-44. Purify biochemical proteins DNA and RNA. Discovery that protein designed enzyme protease and RNA designed enzyme RNAs. The genetic material is DNA. Unique proof that DNA is the genetic material came from the experiment Alfred Hershey and Martha Chich 1952. Virus on the medium that contain radioactive phosphorus and some other mediums that contain radioactive sulfur. Radioactive phosphorus contain radioactive DNA but not radioactive proteins. Sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur. ये हर्सेन चीज का एक्सपेरिमेंट है. यहाँ पे फर्स्ट स्टेज में इन्फेक्शन होता है, सेकंड स्टेज में ब्लेंडिंग होता है और थर्ड स्टेज में सेंट्रीफिकेशन होता है. यहाँ पे फर्स्ट स्टेज में लास्ट वाला जो स्टेज है नॉन रेडिक्टिव 35S सल्फर डिटेक्टेड इन सेल्स एंड रेडिक्टिव 32PS डिटेक्टेड इन द सेल्स. RNA वर्ल्ड. RNA genetic first genetic material है. RNA was the first genetic material. RNA being catalytic was the reactive and has unstable. Replication. Double helix structure of for DNA. Waston and Crick has immediately proposed a scheme for replication of DNA. A possible coping mechanism for the genetic material. Waston and Crick 1953. Each DNA molecules will have one parental and one newly synthesis strand. This scheme was termed as semi-convertative DNA replication. Experimental proof. DNA replication semi-convertedly. It was first in the Escherichia coli. Subsequently, higher organisms such as plant and human cells. Matthew Melson and Franklin Salt performed the following experiment in 1958. They gave E. coli in medium containing 15NH4Cl15N is a heavy isotope of nitrogens. Only nitrogen source for the generation of the result, and the 15N was incorporated into newly synthesized DNA. DNA was centrifugation in a calcium chloride CaCl. 15N is not a radioactive isotope separated from the 14N only based on densities. Second, they transported cell into the medium with the normal 14NH4Cl and took sample various defined time interval. The cell multiplied, extracted the DNA that remained as the double-stranded helices. The various samples were separated independently on the CaCl gradient to measure the densities of the DNA. Third, the DNA that was extended from the culture on the generation after the transform for the 15N to 14N medium. After 20 minutes, recoil divide in 20 minutes. Had hybrid or intermediate density DNA extracted from the culture after the another generation that after 14 minutes and second generation. A very similar experiment involving the use of radioactive thymidine DNA is the chromosomes was proposed on Weiss, Febabena, Tylers and colleagues in 1958. The experiment proved that DNA in chromosome also replication semi-convertedly. The machinery and enzyme. The main enzyme referred to DNA-dependent DNA polymerase, E. coli that has the only 4.6 into 10 raised to 6 base pair compare human whose diploid contain 4.6 into 10 raised to 9 base pair completed the process replication within 18 minutes. Polymerization has approximately 2000 base pairs second. In addition to DNA-dependent DNA polymerase, Small opening of the DNA helix is referred to as replication fork. The DNA depends DNA polymerase catalytic polymerization only in one direction, that 5 to 3 direction. Template with the polarity 3 to 5, the replication continuous, and polarity 5 to 3 direction. It is discontinuous. Discontinuously synthesized fragments are later joined by the enzymes DNA ligase. There is a definite regions E coil DNA where the replication originated. Such as regions are termed as the origin of replication. During the recombinant DNA producers require a vectors. Vector provide the origin of replication. In eukaryotes, replication of the DNA take place S-phase. DNA replication result in the polyploidy chromosomal anomaly. Transformation, copying the genetic information from one strand of the DNA into RNA is termed as the transcription. तो यहाँ पे ट्रांसक्रिप्शन की प्रोसेस है 5 to 3 डायरेक्शन कॉन्टिन्यूस सिंथेसिस होता है और 3 to 5 डायरेक्शन टेम्पलेट डीएनए पेरेंटल स्ट्रैंड होता है और डिसकंटिन्यूस सिंथेसिस होता है. नेवली सिंथेसिस डीएनए 3 to 5 डायरेक्शन 5 to 3 डायरेक्शन रिप्लिकेशन फोर का ये डायग्राम से. ट्रांसक्रिप्शन यूनिट के थ्री टाइप्स है. ट्रांसक्रिप्शन यूनिट इज डिफाइंड प्राइमरली बाय द 3 रीजन्स ऑफ द डीएनए, अ प्रोमोटर एंड सेकंड स्ट्रक्चरल जीन्स एंड थर्ड टर्मिनेटर्स. Since the two strands have opposite polarity and the DNA depend RNA polymerase, polymerization is only one direction, that 5 to 3 direction. Polarity 3 to 5 direction is template and referred to as template strain. Polarity to 5 to 3 direction. Same RNA except thymidine and plus uracil. Do not code for thymidine is referred to as coding strain. तो ये template strain है और ये coding strain है. Promoter, terminator reflects structural genes. A promoter is said to be located toward the 5 end of the off strand. DNA sequence that provide a binding site for RNA polymerase. The terminator is located toward the 3 end downstream. Transcription unit of and genes. A genes is defined as the functional unit of inheritance. DNA sequence coding for the tRNA or rRNA molecules also defined as the genes. Segment of DNA coding for the polypeptide and structural genes. Transcription unit could be said to be monomeric, monocystonic mostly in eukaryotic and polycystonic mostly bacteria prokaryotes. The coding sequence or extracted sequence are defined as exons. Mature or processed RNA exons are interpreted by the introns. Introns or intermediate sequence do not appear in mature processed RNA. Type of RNA and the process transcription. mRNA, RNase and mRNAs, messenger RNAs, tRNAs, transfer RNAs are RNA, ribosomal RNA, mRNA provide template and tRNA bring amino acid and rRNAs plays a structural and catalytic role during the translation. There is a single DNA depends RNA polymerase. RNA polymerase bind to the promoter and transcription initiations. तो ये कुछ प्रोसेस है, प्रोसेस ऑफ ट्रांसक्रिप्शन इन बैक्टीरिया. फर्स्ट प्रोसेस है आपका इनिटिएशन, सेकंड इलोंगेशन और थर्ड टर्मिनेशंस. The RNA polymerase 1 transcribed rRNAs, the 20S, 18SS and 5.8SS. RNA polymerase 3 is responsible for transcription of tRNA, 5 srRNA, snRNA, small nuclear RNAs. RNA polymerase 2 transcribe precursors of mRNA, heterogeneous nuclear RNAs. Exons and introns are non-functional. Subjects process called skipping. Introns are removed, exons are jointed. H and RNA capping and tailing. Capping and unusual nucleotide methyl guanosine and triphosphate is added to fine end of hnRNA. In tailing, adenine residues 200 to 300 are the added 3 and template. RNA molecule with defined combinations of bases, homopolymers and compolymers. Marshall-Nichols cells free system for protein synthesis. Servo-Okaza enzyme polynucleotides polyphosphorylase. Silent feature of genetic code are as follow. First, the codon is triplet. 61 codon code for amino acid and 3 codon do not code for any amino acid, hence they function as stop codon. Second, some amino acid are coded by more than one codon, hence the code is degenerated. Third, the codon is readed in mRNA and continuous fissions, there are no functionations. Fourth, the code is nearly universal. For example, for bacteria, humans, UUA will be the phenylalanine. Some expansion to these rules have been found to mitochondrial codon and some of the protozoans. AUG, the dual function, it is the code for methionines meet at in the also inhibitor codon. UAA, UAG, UGA are stop terminator codon. Mutation and genetic code. The mutation effect on the point of mutations. A classical example of point of mutation is change in the single base pair in the genes for beta globin, change that result in the change in amino acid and result in glumatic to valenines or result into disease. The condition is called sickle cell anemia. इसे हम इस कंडीशन में पढ़ते हैं. इस कंडीशन की तरफ बीटा ग्लोबिन चेंज जो है चेंज होता रहता है. ट्रांसलेशन. Ribosome also acts as the catalytic 23S RNA in bacteria is the enzyme ribosome for the formation of polypeptide bonds and start codon AUG, stop codon code for polypeptides and mRNA not translated and referred to as untranslated regions of UTR and UTR present in the both the 5 and before start codon, 3 and after stop codon. Ribosome bind to the mRNA start codon AUG only by the inhibitor tRNAs. Appropriate codon in mRNA by the forming complementary base pairing with the tRNA anticodons. रेगुलेशन ऑफ जीन एक्सप्रेशन. रेगुलेशन एंड जीन एक्सप्रेशन यहाँ पे 4th स्टेज में डिवाइड करता है. ट्रांसक्रिप्शन लेवल फॉर्मेशन ऑफ प्राइमरी ट्रांसक्रिप्ट एंड सेकंड प्रोसेसिंग लेवल रेगुलेशन ऑफ स्प्लिपिंग एंड थर्ड ट्रांसपोर्ट mRNA फ्रॉम द न्यूक्लियस टू द साइटोप्लाज्म. फोर्थ ट्रांसलेशन लेवल. Enzyme called beta-galactoside is synthesized by the E. coli. It is used to catalyze the hydrolysis of disaccharide lactose into galactose and glucose. Bacteria do not have lactose around them, do not utilize for energy source. Synthesis of enzyme beta-galactoside. Regulatory protein can be act as the both positively activators and negatively repressor. In interaction of protein with sequence stromas operators. Operator bind to repressor protein. Lack of operator is present only the lack of operands. Lack of operands. Genetics Francisco Jepko and biochemist Jepko Mode. The first elucidated transcriptionally regulated system lack operands on lack refer to lactose polycystonic structural genes in regulated by the promoter and regulatory genes. In bacteria and refer to as operand. Lack operand consists of regulatory genes in the I genes. 3 structural genes Z, Y and A. I gene code for the repressor of the lack of operand. Z gene code for beta-galactoside B-GALAX and responsible for hydrolysis of disaccharide. Y gene code for the primers increases permeability of the cells of the beta-galactosides. A genes code for translatolysis and lack operand required for the metabolism of lactose. Human Genome Project. Human Genome was launched in 1990. Human Genome Project HGP was called mega project. Approximately 3 into 10 raised to 9 base pair sequence require $$ into 3% of the base pair. Human Genome Project $9 billion used with approximately beginning project will approximately $9 billion. Each page of the book contain 1000 letter. Each book contain 1000 page and 3300 such as book will be required store information of DNA sequence for the single human DNA cell. यहाँ पे कुछ गोल्स है ह्यूमन जीनोम प्रोजेक्ट के. SCP goals, some of the important goals SGP were also follow. Identify all the approximately 20,000 to 25,000 genes in human DNA. Second, determine the sequence of the 3 billion chemical base pair that make up the human DNA. And third, store this is information is database. And fourth, improve the tool for the data analysis. And fourth is fifth is the transport related technology to other sectors and such as industries. And sixth is the address the ethical and legal social issues that may arise from the project. Human Genome Project, the 13-year project, and US Department of Energy, National Institute of Health. During the early years, HGP well trust UK become major partner. Additional contribution Japan, France, and Germany, China, and other project complete 2003. Applied toward the slowing challenge of health and care, agriculture, many of the non-human made model organisms such as bacteria, yeast, and Conebellicis, insects, free-living, non-pathogenic nematodes, Drosophila, and fruit flies, and Arabidis. Methanogens, RNA referred to a sequence take ETS. The term referred to a sequence of annotations. DNA enclosed suitable host using the spacing vector, calling result amplification of each species DNA fragment so that it subsequently could be sequenced with the is the used host where bacteria est and vectors were called VGA bacterial artificial chromosome, YCA est artificial chromosome. Fragment were sequential using the autonomous DNA sequence that work on the principle of method developed by the Franz Singers. Sequence of the chromosomes first was complete only in May 2006. Last and most important parts. DNA fingerprinting is very quick way to compare the DNA sequence of any individuals. DNA fingerprinting involves the identifying difference in some of the species regions in DNA sequence is called repetitive DNA. A sequence small stretch of DNA repeated many times. The bulk of DNA from the major peak and the other small peak is referred to as satellite DNA. Depending on the base composition A, T, rich, G, C, rich. Satellite DNA is classified into many categories such as microsatellite, mini-satellite. This sequence normally do not code for any protein, but they form large portions of human genomes. This sequence show high degree of polymorphisms and from the basis of DNA fingerprinting. DNA from the every tissue such as blood, hair, follicles, skin, bone and saliva and sperm. Polymorphism inheritable from the parents to children. DNA fingerprinting is the basis of parental testing and in case of the dispute. A polymorphism DNA sequence is the basis of genetic mapping and human genomes with us DNA fingerprinting. It is essential that we understand what DNA polymorphism means. It is simple term polymorphism. Variations in genetic level tissue do not mutations. A name mutation may arise in individual either somatic cell or in the germ cells. Sequence variation has traditionally been described as DNA polymorphism is more than the one variant allele and locus occur in human population with a frequency greater than 0.01. The technique of DNA fingerprinting was initially developed by Al-Jeffrey. He used satellite DNA as probe that show very high degree of polymorphism, variable number of tandems and repeat. इसको हम क्या कहते हैं VNTRs. Involves southern blot hybridization using the radio labeled VNTR probes included first isolation of DNA, second digestion DNA restriction endonucleases, third separation of DNA fragment by the electrophoresis, and fourth the transforming blotting and separated DNA fragment to synthesize membrane such as nitrocellulose or nylons, and fifth hybridization using labeled VNTR probes, and sixth is the detection of hybridization DNA fragment by the audio radiographies and schematic representation of DNA fingerprinting. VNTR belong to class satellite DNA referred to as mini satellite. VNTR varies size from 0.1 to 20 kbs. Hybridization is VNTR probes. Population expert in the case of the monozygotic identical twins. Many different probes are used to generate DNA fingerprints. तो यहाँ पे हमारा चैप्टर ख़त्म होता है. ये चैप्टर उन स्टूडेंट के लिए नहीं था जो पूरा की पूरा चैप्टर पढ़ना चाहते थे. ये लेक्चर उन स्टूडेंट के लिए था जो इंपॉर्टेंट पॉइंट्स के बारे में पढ़ना चाहते थे और सारे के सारे जो इंपॉर्टेंट क्वेश्चन आते हैं इस चैप्टर से वो पॉइंट पढ़ना चाहते थे. उन स्टूडेंट के लिए था ये लेक्चर. तो अगर आपको ये वीडियो अच्छा लगता है तो सब्सक्राइब जरूर कर देना है और अपने दोस्तों को शेयर जरूर कर देना है.